A.Gutierrez. She's a biology major, and I'm going to let her tell you her topic because I can't pronounce most of it. And see if it turned on.
It is, yeah.
It's
it is.
nice.
So, as Dr. Minor said, my name is A.Gutierrez-Cano, and today I'm going to be presenting my research on using UV mutagenesis to produce ethanol-tolerant mutants of Saccharomyces cerevisiae 09448. Okay, this was not fun. I'm a biology major. Okay. So, for all of you who are wondering why the summers in Texas are getting so hot and why we're starting to experience more winter storms, the short answer is because of global warming. Global warming is caused by an excess release of greenhouse gases into our atmosphere, which act as a blanket for the planet that the planet can't take off. And blankets doing as blankets do, it increases the surface temperature of the planet and hence causing global warming. And a human activity such as transportation will contribute to global warming because of its near complete dependence on petroleum, which is a fossil fuel that is used to make the gasoline that powers our cars. So as can be seen in this diagram, the drilling, processing, shipping, and then the burning of these fossil fuels emit carbon dioxide into our atmosphere, which is an example of greenhouse gas and contributes to global warming. And an alternative to a fossil fuel, which we hope will become more prevalent in the future, is the use of biofuels, which in contrast to fossil fuels, it works in a cycle, and the cycle will start with the farming of plants, and these plants that are farmed are transported to a fermentation processing center where they're used to produce ethanol. And the ethanol that's produced from these plants will replace the petroleum found in the gasoline for our cars, and this is not to say that whenever we burn the ethanol there isn't any carbon dioxide released into the atmosphere, there is. But instead, the carbon dioxide that's released from the burnt ethanol will use to grow the plants that will later enter the cycle. So it's almost a net zero release of greenhouse gases into our atmosphere and not contributing as much to global warming as fossil fuels would. And as I mentioned in this step, the farm plants have to be used to produce the ethanol, so you can't just shove corn down your gas tank. I would not suggest that. You won't get very far that way. But instead, the corn has to first be fermented. And this fermentation process is carried out by microorganisms like yeast who, like you and me, use sugar for energy. And so in order to get these two molecules of ATP, which is their form of energy, they produce ethanol as a result using glycolysis and fermentation. So now that we've kind of dove a little bit into the fermentation process, let's learn more about the biofuels that are produced. So we produce biofuels already. The catch is, though, we don't produce enough for us to see a significant impact on our climate. And this is because we rely heavily on what's called first-generation biomass, which is corn, for example. Corn, or first-generation biomass in general, is really easy to use because, as you can see in this diagram, it's made up of mainly starch, so it's really easy to ferment and really inexpensive to ferment. But as I've said, we don't have enough of it, so it's causing us to kind of look to alternatives that we can use, such as second-generation biomass. And a more specific example of second-generation biomass is pectin-rich biomass. And apple pomace, citrus waste, and sugar beet pulp, as you can see, is a lot more complex than corn. And so the reason we don't already use pectin-rich biomass is because of how complex the cell walls of these plants are. So this process of using second-generation biomass is expensive because we have to add enzymes to break up these really complex molecules of pectin in order to get these single sugars that we can then feed the yeast to produce ethanol with. So fortunately, though, we didn't discover it, but a yeast strain was discovered called Saccharomyces cerevisiae 09448, which makes its own pectinase enzymes that can cut up those complex molecules. So using this strain would be more affordable and less expensive than using our already used industrially prepared yeast strain and also have to add the enzymes on top of it. So this part makes it more expensive, so we're hoping to use 09448 in the future. Unlike ADY or active dry yeast, which is already used industrially, 09448 can't withstand the industrial conditions such as the increased temperature, the increased osmotic pressure, the decreased pHs or just the more acidic environments, and the increased ethanol concentrations like ADY can. So in our lab, we're trying to see how we can get 09448 to withstand those industrial conditions like AUI. And so in our research specifically, we wanted to see how we can get 09448 to withstand those higher ethanol concentrations that it'll be exposed to later. And the way we decided to approach this issue was using UV mutagenesis, and we got this protocol from a paper we read like a couple summers ago, where Saccharomyces cerevisiae BS1 and BS3 were exposed to UV radiation, and the mutants that were produced from that radiation were showing higher ethanol tolerance, osmotic pressure tolerance, and thermotolerance. And the way this UV mutagenesis works is UV radiation, like the radiation that comes from the sun, causes DNA damage, and that DNA damage will lead to mutation. So that's why you get, like, some people get skin cancers because this DNA damage is caused by UV radiation. So our hope is that if we expose our 09448 to UV radiation, we could get mutants and hope that some of those mutants will acquire mutations that allow them to be more tolerant to ethanol than the unmutated version of 09448. So that leads us to our research question: can we generate ethanol-resistant strains using a UV mutagenesis protocol? So now into our methodology. This whole part of the methods is like us preparing the 09448. And once we prepared 09448 for UV exposure, we divided those samples of 09448 into 7 groups. So the first group wasn't exposed to any UV radiation. The second group was exposed to 30 seconds of UV radiation. The third group was exposed to 60 seconds, so on and so forth, until we had our 7th group, which was exposed to UV radiation for 3 minutes. Then once we had our mutants, we grew all of those samples that were exposed to UV radiation on TSA plates that had 10% ethanol in order to see, of those cells that were mutated, which ones were already showing some level of ethanol tolerance that 09448 couldn't show. So once we had some cells growing on these ethanol plates, we organized them onto what's called a patch plate. And then from these patch plates, we chose 12 mutants at random for comparison with our unmutated version of 09448. So in this first column of what's called a 96 well plate, we exposed our control to 0%, 2%, 4%, and 6% ethanol concentrations, and then every column after that is a different mutant that we wanted to compare to. We put this 96 well plate into a plate reader, which would measure the optical density or the growth of these mutants and the control when exposed to these ethanol concentrations for over 36 hours, measuring the optical density every 15 minutes to build a growth curve. So what's a growth curve, but more first, what's an optical density? An optical density value is representative of the number of cells found in that sample. And it's measured using light. So the plate reader will shoot a beam of light through a sample, and it gives us an optical density value based on how much light reaches the detector on the other side of the plate reader. So for example, say we just put a sample into our plate reader, and there's not really much growth yet, so the plate reader will shoot a light through the sample, and all the light will pass through the sample and reach the detector. And since all the light reaches the detector, it's a signal that there's not really many cells in that sample, and so we get a lower optical density value because of it. Say maybe 10 minutes later, we're starting to see a lot more growth in the sample, and so the light that's shot by the plate reader and runs through the sample will be reflected off of the cells, and then not much light will reach the detector as a result, and therefore that's a sign that there's more cells in that sample because there's not much light reaching the detector, and we get a higher optical density value. So we measure these optical density values, or we plot them on just a simple graph. And so say here's right whenever we put into the plate reader, the optical density is still low. 15 minutes later, the optical density is still low, there's not really much growth yet. But say maybe an hour later, we're starting to see a rise in the optical density values. And so we keep plotting this until we get what's called a growth curve. And what we're most interested in is the highest optical density that those samples will reach, because it's a sign of the max number of cells that we're able to grow in that ethanol concentration or the environment that it's in. So we built a growth curve of the control and our 12 mutants and compared them to one another in 0%, 2%, and 4%, and then also 6% ethanol. And as you can see, in our 4% and our 6% ethanol, we're starting to see a lot more divergence, or the mutant's capabilities of growing in those environments. And so we kind of turned this into a bar graph so we can see the final optical densities that were reached a lot easier. And what we're interested in is, again, the mutants that were able to reach a higher optical density than our control. So in 4% and 6% ethanol, the mutants that were able to reach a higher optical density than our control was our mutants 1, 2, 4, 6, 7, 9, 10, 11, and 12. We also looked at the generation times of the mutants compared to our control, and a generation time is the amount of time it takes for the population to double. So let's say we have an orange group and we also have a blue group. And we're interested in how long it takes for these 3 cells in each of these populations to turn into 6 cells. So let's say it takes the orange group 2 minutes to turn from 3 to 6, and it takes the blue group 6 minutes to turn from 3 to 6. That means the orange group has a shorter generation time and therefore grew better in that environment because oranges could withstand those ethanol concentrations better. So we created a bar graph of the minimum generation times, and again, we're interested in the mutants that have shorter minimum generation times than our control. So it's really clearly evident that mutants 3 and 8 do not have generation time shorter than the control, and it's actually skewing our results, or the way we can see this graph a little bit. So we created another bar graph, not including 3 and 8, and now we can also see that mutants 5 and 10 also do not have generation time shorter than the control, so they wouldn't be considered our mutants of interest. So taking these 2 factors into account, the mutants that were able to show greater final optical densities in ethanol concentrations 4 and 6 and have shorter minimum generation times than 4% and 6% ethanol concentrations were mutants 1, 2, 4, 6, 7, 9, 11, and 12. So in conclusion, we were able to find or get 8 mutants that out of the 12 that we had experimented with that were showing higher ethanol tolerance than our unmutated version of 09448. However, in the future, because currently we only did this experiment once, so we needed to do these experiments again and create replicates to see if we would get consistent results. We also want to see if these ethanol tolerant mutants can also withstand the higher temperatures that they encounter, higher osmotic pressures, and more acidic environments that they would see if they were used industrially. But if it turns out that these 8 mutants, or at least some of these mutants, are able to withstand all those industrial conditions, if they could be used on a large scale and undergo large fermentation using pectin-rich biomass, and overall decrease the impact that our transportation has on global warming. I would like to thank Dr. Edwards for organizing our research, as well as Jennifer for helping me collect this data, and Jordan Macaria and Bryce Jones for helping standardize the protocol that we use, as well as St. Edward's University, Department of Biological Sciences, the Ronald E. McNair Post-Bachelor Achievement Program, and the I4 Institute for funding. Thank you. That's a good one. I think it was coming to, because we had to, the UV part where we were exposing them to UV radiation, we had to do that like 3 times because like we accidentally switched the numbers in our calculations, or we used the ethanol plates that we had used were just not prepared correctly, so that we weren't just getting any growth on them after we exposed them. And so it was kind of coming to just the acceptance that sometimes we'll have to do experiments over again, and that's okay. Like, we'll learn from our mistakes, and we perfected it over time. So I think just being okay with having to do those things again, and being all right, like it's not going to happen right the first time. Probably that was the hardest part, but I've also come to enjoy it more because I like learning from my mistakes now.
So for the 8 mutants of interest, like what would be your next steps for getting them to tolerate industrial sort of environments?
So after we, because I think, like it was weird because like, here, I want to show you this. Okay, so can you see like how some of them had like short minimum generation times at 6% than they did at 4%? So that would be like they're more tolerant under higher ethanol concentrations than the 4%. So we want to do these experiments again to see if that's like, if that really is the case and why that is the case. But then to see if we just also want to see if they can like grow under like higher temperatures, for example, or higher osmotic pressures. And if they can do that, I don't know this about that. I don't have to talk to my PI about that one. But we first have to see if they can even do those things in general. And also the industrial conditions that they have to be able to withstand, like, is like 15%, and we only expose them to 6%. So we still have a lot more to adjust into, but we'll have to see.
I know you've done research in the past.
Yes.
How was this research different than your previous research? If
Mhm.
it was, and what was what did you enjoy about this project?
Okay, actually, these are really good questions because I love the research that I do so much. So previously, so this is new, we just recently started using mutagenesis, but whenever I did research with Dr. Evans before, did directed evolution. So we were just slowly increasing like ethanol concentrations on 09448 to see like if over time, it's called directed evolution. So like whenever you expose an organism to a stressor, you're hoping that like the ones who have the characteristics that you want will survive, and the ones that don't have those characteristics that you want will die, and so you're left with only the ones that have the thing that you want, that tolerance. And so over time, we were slowly increasing the ethanol concentrations for 09448 and just hoping like whatever cells were able to survive that ethanol concentration they're previously in could also withstand more intense ones. So that process took a long time just because like it was, sometimes they didn't acquire the mutations that you needed. And I recently also rediscovered the research that we had did, and there was like a month where we weren't seeing any results, but just because the incubator that they were growing in wasn't reaching the temperature that we thought it was at. So it was like the whole month was just nothing. It was crazy. But yeah, I think this one is faster. I like it more, but like, I don't know. I think all of it's cool.
Any other questions?
Oh, my God.
Drastic increase in electric vehicles and transportation. How do you see that fitting in with industrial applications?
Ooh. I don't know. So I know that like just because things are said that they're electric, it doesn't mean that they are just purely electric, because like the electricity that powers our houses still require like fossil fuels to be burned in order for that electricity to be sent into our homes. So there's still some fossil fuels being used to get that electricity to our house. So like say you charge your Tesla at your house, like that energy, that electricity that you use to charge your Tesla is still using fossil fuels in the background. So I still think that this would be useful even for them, because if you use biofuels overall, that electricity that you're giving your car will, in the background, like still be better for the environment. And,
Yes,
sorry. I got a little passionate.